So let's welcome Sunanda Sharma up to the stage here so we can see what's behind all that. Thank you so much for joining us.
Cool. Thank you. So yeah, Joey earlier today talked about the microbial communities that exist within each of us, and he sort of introduced the concept of symbiosis, which I'm sure many of you have heard about. The idea is that the microbes that live within us, they have a sort of mutualistic benefit. We provide a home for them in our gut, or actually on our skin and other places as well, and they provide us benefits as well. And that's an example of symbiosis. And that area of symbiotic design is something that I've gotten really interested in, and a lot of people at the Media Lab, specifically in my group, are interested in that as well. So Patrick talked about design on the very small scale and on another scale as well. We're talking about design a little bit in the more stereotypical sense, so designing products and designing architecture. Yeah, so I'm part of the Mediated Matter group, so I'm doing my PhD right now. And I came from a background of biology, but like many other people, I saw that biology could relate to design, or rather, I was told by so many people that biology and design are related, so I should get in there as soon as I can. And now I have the chance to work with people of all different disciplines. So as Joey mentioned, we're in a sort of anti-disciplinary environment. So we have people on our team who are architects, like my professor, Neri Oxman. We have designers, we have engineers, and biologists, like myself. So our work is all about designing for, with, and by nature. And it's at the interfaces of several different fields. One is fabrication, materials, computation, and most recently, biology. And all of this still falls under the umbrella of design, because that's sort of the lens through which we like to view each one of these fields. So in most of our projects, we try to design across scales, as Neri calls it. So a lot of this begins with the exploration of a single organism. So shown here is a silkworm. And by studying the behavioral dynamics of a single silkworm, we were able to figure out how to collaborate with 6,500 of them to make an architectural scale pavilion, which was part spun with machines, and the rest was filled in by the silkworms over a period of 3 weeks. And so this was demoed in 2013 in the Media Lab lobby. Another example is looking at materials. So we looked at the material that's in a shrimp exoskeleton, so the shell of any sort of crustacean, has a material called chitin. And if you deacetylate it and then you add water, you can get a hydrogel made of chitosan. And we discovered a way to 3D print this to make large-scale scaffolds that are also biodegradable. So if you put them back into water, they'll expand and swell. And we think it could be a new, interesting scaffold instead of things that are less green. So for the project I'm talking about today, the organism in this case are 2 different types of bacteria. So shown here is one of them, E. coli. So one of the first questions people ask whenever I bring up this topic is, why living wearables? Why would you want your clothes to be living or breathing? Why not just have shirts the way we have them right now? And Patrick definitely talked about this a lot. So biology has a lot of benefits, and that's sort of why I think designers are interested in biology, and then it leads to why biologists are interested in design. So there are certain characteristics that unify all living systems, and the list changes depending on who you ask, but the ones that I've included are homeostasis, adaptation, reproduction, growth, metabolism, organization, and responsiveness. And then there's other things that were mentioned earlier as well, which is sensing, self-repair, feedback, so amplification and dampening of signals, and complexity, such as in hierarchical organization. So protein folding, for example, is a really good instance of this. And so if you have products that also have all of these sorts of qualities, then you start getting a dynamic sort of feedback loop, and you can mediate between humans and the environment. And then biologists, I think, are starting to be interested in design because it affords us the opportunity to take all the things that we've done inside the lab, so that usually stays within petri dishes or flasks, take it outside of the lab and to find new scales and contexts for which to apply them. And then, as I said, that dynamic mediation, especially on the wearable scale between the human and the environment, can then be done. So when we first started thinking about bringing living things out of the lab, the first question was how to contain them, because you want this to be done in a controlled way. So a few years ago, we started looking at microfluidic systems. So for some years now, we've partnered with Stratasys, which is a 3D printing company. And shown here is a valve, a chip that we had made that can handle very small volumes of liquids. So microfluidics is interesting in any biolab because that's how you pipette things. So basically, being able to control small volumes is important because you can use them in DNA assembly, for example. And by using a multi-material printer that has rigid and soft components, you can make pumps, you can make valves, and you can control that liquid flow very well. And as most things in our group, this was scaled up. So we started looking at meso and macrofluidics. So shown here are some first potential renders for what larger scale products would be like that also have internal channels. And the reason channels were of interest to us is because then you can control living liquid cultures of organisms. So then the next step was coming up with some potential renders for what a wearable that has fluidic channels would even look like on the body, because there's not many that exist. And this led to the creation of a series of wearables, and these are the first 3 of them. And this was part of a collection called The Wanderers. So as you can see, each one of them is sort of reminiscent of or inspired by a human organ system. So for example, the one on the left is inspired by the brain. So you can see sort of the sulci structure there. And the 4th one is the one that we explored more, and the piece is called Mushtari. And so as you can see, it looks sort of like a gut, which is exactly what it's supposed to look like. It's supposed to be an external gut. So your internal gut, as we've talked about, takes in food and then extracts the nutrients that your body needs in order to survive, and then also has all these other implications with your mind or your behavior. And we thought creating an external gut from scratch would maybe allow us to understand more and then take a different sort of approach, a sort of reverse approach versus what most people are doing right now. So this is the piece that's worn on the hips. And the entire thing is 5 pieces, 5 printed pieces, which when you put them together, they make 59 meters of channels. And so this is a video that I think some of you have seen. It'll go over everything generally, and then I'll go more in detail after that. Yeah. So that was just the overview of the project. And here's sort of a zoomed-in look at the internal channels there. So you can see there's a gradation in optical clarity. So there's the completely transparent parts, and then there's the parts that are more opaque. And through that gradation, that's how we wanted to control the symbiosis between two different cultures of organisms that lived within this piece. So these two types of organisms were, one was cyanobacteria, which is, it's a model organism that many people know of, and it's been referred to as a microbial factory because it's been engineered to produce many different things. For example, sucrose, like the strain that we used here, but also it's been used to create biodiesels and ethanol and other sorts of fuel. And for this, we partnered with Pamela Silver's lab at Harvard. And the second culture was E. coli. So E. coli is one of the most common organisms that you can find in a lab, but also in the real world and in your gut as well. And E. coli has also been engineered to produce many different things, including insulin and many other sorts of therapeutic proteins and antibiotics. And so we were thinking of this sort of piece, this fluidic wearable, as a platform technology. So you can switch out the different types of organisms, but then have the same system and try to mediate how each of the organisms relate to one another through how the material distribution is made. So this is sort of what the final piece looked like, as you saw in the video. So since then, we've moved past just looking at bacteria, and now in our wet lab, which is what we call a designer's wet lab, it has organisms from all the kingdoms of life. And so the wet lab itself was an interesting area because that's how we sort of tried to design symbiosis between designers and between scientists. So this is one of the initial renders of the space, which was then completed in January of this year. So this is the first Biosafety Level 2 space in the Media Lab, which was a big deal for us because so many of us are now interested in biology. So as you can see, it doesn't look like a typical space because we're trying to think about it as both a display area and a place where you can do hardcore research. So having transparent glass walls is not something that most BSL-2 labs usually have because people don't think that the public wants to see that, and there's sort of a shielding that happens there. But we're trying to break that. In the Media Lab, all the walls are clear because we want everybody to see the research, and we want the public to engage with what's happening within our labs. So yeah, that's pretty much everything that we're doing right now. Thank you, and thank you to Digital Garage.
Wow. I said wow beforehand. I'll say wow again. Thank you, Sharma-san. Holy smokes. This day is getting weirder and weirder, but in a good way. Thank you guys so much for sticking around. And next, we have someone who's also taking us into the wearable realm of biology. I mean, I'm wearing biology right now, but then I put on this other stuff to kind of cover it up. I hadn't thought about wearing my guts on the outside. That's so fun. The next person has a PhD in chemical and pharmaceutical engineering. And she's a research scientist at the MIT Media Lab, but also the MIT Department of Chemical Engineering. So people are getting freaky with chemicals and liquids and biology and stuff. So this person merges biotech, advanced materials, and design to do biofabrication and biomanufacture, which I feel like after today I'm going to call up my mom and be like, Mom, I learned what biomanufacturing and biofabrication are. Please come tell us, Wen Wang.
We live in a symbiotic way where microbes are everywhere, in the environment, on our body, and even inside of our guts. So you might be wondering, what's the relationship between microbes with these beautiful artistic wearables? And how we made them, and what's the function? So now I would like to walk you through the journey of garment development and also my research life. So the journey starts from my hometown in Mongolia. I was born there and grew up in this beautiful place where people live in harmony with nature. I'm always amazed by how powerful nature is, especially for this biological system, how it interacts with the natural environment. Especially when I was young, I liked to pick up pine cones because it can transform at different humidity conditions, and this way helps the pine cone to spread the seeds into the nature. So I wonder how it works and how can we utilize this way to make something that is useful. And so that's why I chose bioengineering as my major during undergrad and continue the research during PhD and postdoctoral research at MIT. So during these years, I work on different types of systems and trying to unveil the fact how nature transforms chemically. So I developed normal strategies of making sustainable chemicals and also biofuels, and also some cheaper biopharmaceuticals to enhance our well-being. However, all these useful materials, it can be produced as a raw material. We still need to go through very tedious post-processing to make it into the form that general public can consume, can understand. For instance, to make a cancer therapy drug pill, a capsule, after you make this functional intermediates, you still need to go through tens of steps to do the post-fabrication and formulation. So I was wondering, can we change this? And what's the way to do it? So can we just culture a mushroom that can produce some use for cancer therapy drugs in our own kitchen? And we don't need to go through all this process, wasting all this energy and water. Or can we just engineer a plant that has the biofuel in its vein, and you can connect the plant with maybe your car tank and try to fuel the car. Try to avoid how maybe we can remove the steps of bioprocessing from the biomass to bioethanol and biodiesel. Or maybe can we grow a clothes in our own wardrobe, and the clothes can understand us, understand the skin condition, and also, we don't need to use all this crude oil-derived chemicals. In order to do that, we need to shift our focus from understanding the biologic activities to the materiality, to really consider all this biomass as a material, instead of just consider it to do the functionality. And we need to think about how we can use it to build something out. To start this journey, I thought microbes must be a really good option to start with, especially natto cells. You have heard a lot this morning about the magic thing about natto bacteria and how it can enhance our health. I choose natto bacteria because it can grow really fast. Because the formal name is called Bacillus subtilis, and we have ready-to-use genetic tools. So everything is perfect. And we start to look into the materiality of natto bacteria. So it contains 70% of water and 30% of other type of chemicals, including proteins, DNA, RNA, and polysaccharide. So it's very similar to all other kinds of biological materials, including pine cones. So I was wondering, is it possible to realize my dream using this natto bacteria? Maybe that's might happen. So I start to put the natto bacteria at different humidity conditions. What I've observed is that the natto bacteria can actually shrink at dry condition and swell at humid condition. So this shrinking and swelling behavior can help us maybe do some further engineering on making a structure of a material and then making a system that can sense the environmental humidity just as pine cone did. So we came up with this composite structure that has the natto bacteria on the top, and on the bottom we have a non-functional natural latex or other type of fabrics. And the magic moment comes that we put this film under different conditions. When it gets dried, it shrinks, and then it starts to bend. At humid condition, it flattens down. So this transformation happens really fast, like within a few seconds. It's instant. Such as if you breathe on this fabric, it will change its shape and change how it behaves. So with this tool, we start to working at different type of design, such as one-dimensional shrinking, two-dimensional folding and expanding, even three-dimensional origami structure change, or change the surface texture of the whole material. So this provides us a great space to come up with new ideas and utilize this material to make some really interesting objects, such as this very cute tea bags. It has biohybrid tea leaves on top. So when you put the tea into it, it can sense the humidity change and can tell you when your tea is ready. Or these beautiful flowers that when you spray water on it, it can sense the humidity change and change both color and shape at the same time. So it's like the real flower blossom. Or this living lampshade. When you turn on the light, it senses the temperature change from the light bulb and also can change the shape and give you different shadows. So more importantly, we are working with living organism and want to utilize for our body. So this is about utilizing our own sweat to trigger the change of the garment. So I would like to invite Colleen on the stage. So she is wearing the bacterial garment now. So we want to show the bag as well. So the garment is, thank you, yeah. So the garment is made of bacteria, and it can sense the change of the humidity. So when you start to sweat, the garment can open up automatically like a fish scale. So in this way, it can help you to modulate your temperature and your body sweatness to make a better, maybe make your skin much comfortable. So we start this journey by in the wet lab. So culture the bacteria in a bioreactor. So bacteria can grow really, really fast. The doubling rate is 15 minutes. That means from one cell, overnight, it becomes trillions of cells. So the bacteria is very tiny. It's only about 1 to 3 micrometer in length. So we characterize the bacterial behavior through atomic force measurement, where you can see that the bacteria, it changes its shape and volume at different relative humidity conditions. So by utilizing this technology, we are able to make a garment through digital printing, transferring the solution into a printer and extrude it out at the place we want to extrude and the amount. We can have an accurate control of the performance of the garment as well. So this shows how it gets produced. So the printer system is also very versatile. So we can control the flow rate, we can control where to deposit and how much we want to deposit. So after depositing the bacteria, the whole garment becomes alive. So it's not a dead garment on our body. It can tell you how healthy you are and also help you to be more healthy and help you to really release all your sweat away from the body. So we also developed a parametric model considering both the heat map and the sweat map on our back and optimize the whole distribution of the sweat. So the garment itself is alive. So the dancers from Boston Ballet Company, when they wear the garment and start to dance, they felt like they want to create some really good movement to show their back instead of the arm. And also we got contact with 2020 Olympic ambassador in Japan, thanks for Joe Ito. And he's also interested in our work. Probably after a few years, you might see some volunteers or even athletes wearing this beautiful garment. So the work is a collaborative work with multiple departments from chemical engineering to mechanical engineering to biologic engineering, and also Royal College of Arts. So we hope that in future the garment can really be something that everyone can have this garment and can enhance the healthiness and the well-being. Because I'm from MIT, so we like math. Beyond the artistic part, we also did very in-depth research on how this garment transforms. So this is a mathematical equation describing the process and the simulation to see how much force it generates when it's bent. And in future, we also want to combine genetic engineering with the garment, such as make the garment glow at the dark. So these are some preliminary research to show that at a different humidity condition, we are able to control how bright the garment is by combining some fluorescent proteins. So we are human beings, but we are not alone. We have all kinds of microbes on our body, inside our body, and we really hope that by using this way, making the technology more accessible to the general public, they can understand more about bacteria and about the microbes. And also, maybe in future, instead of engaging them to only the garment, but also maybe making the garment that can influence the environment, such as the garment that can absorb those carbon dioxide or maybe aldehyde from the environment to really purify the air. Or maybe a garment that doesn't need to be washed anymore and can release out all the really good-smelling fragrance after your gym. So we hope to combine science, design, engineering, and also arts. So recently, we were invited to go to the Centre Pompidou next year. So next March, hope you can visit Paris and see our exhibition. So as Joe Ito mentioned, bio is the new digital. So we believe so. bioLogic is part of it, and we hope to make the world better. So last but not the least, I would like to thank the whole team from Tangible Media Group at MIT Media Lab, and also all our collaborators, and Hiroshi Ishii as our PI. He's really inspiring, and Joe Ito for inviting us to join this wonderful talk. And thank you for Digital Garage to give me this opportunity.
Thank you so much. Wait, will you stay up here for one moment? Colleen, will you come back up here for one moment? You called her up here, you got her in this cool outfit, and then you're like, okay, thanks. So you're saying that this thing has been 3D printed with like bio bits? And
yes, so we use a printer to print these bacterial flaps on top of it. So you can see that all these small flaps, it can open up when you start to sweat. So I actually wear it during the gym by putting temperature and humidity sensor on my skin, measuring the heat generated and also the sweatiness. So it shows that it can really help me to cool down my body. And all these flaps, after you start to gym, it all open up. But once you remove it, it becomes flattened very fast. So the response time is like within a few seconds.
That's super cool.
Yeah, imagine it's like an air conditioner on your back.
Will somebody take Colleen out dancing tonight and take some photos so we can...
She's already worried I'm going to steal this.
But thank you so much.
Yeah, thank you both so much. Yeah, wow.